So the second part of this video series, now that we've learned about how fuel metabolism happens with glucose, we're going to map on how it happens with proteins and then with fats, and then think about how those dovetail in with stuff that we've already learned about. We'll kind of have a few pieces to sort of wrap up some of the details of fuel metabolism, and then we'll close out here with an understanding of how pKa works, which is sort of this master protein phosphorylator. Okay, fat metabolism here. So like we've seen with glycolysis then in gluconeogenesis, we're going to see opposing pathways that happen in catabolic versus anabolic states. So when we think about fat metabolism, and again, we're talking about the metabolism and the breakdown of fatty acids, the net is that we've got a fatty acid that is found its way into the mitochondria. In a 4-step process, it's converted into acetyl-CoA. So there's a acetyl-CoA little cartoon down here. So each of these little spheres represents, again, a carbon atom. That's been the consistent theme with these pictures that we've shown. And we've talked about what you do with acetyl-CoA in the liver depends on whether you're in a need state or a fed state. In a need state, and this is, again, a local need state, well, I should say, let me back up here. If we're talking about acetyl-CoA in a need state, you actually differentiate in the liver whether it's a local versus a global need state. So if the liver just needs its own energy, it will take that acetyl-CoA and take it onto the citric acid cycle. But mapping on here, if you are in a global need state, the liver can help with that by taking acetyl-CoA, zipping 2 of them back together to make ketone bodies. We'll talk about those on the next slide. And ketone bodies are a hydrophilic circulating fuel that can be used by all cells to kind of get energy. So it's a way, remember, if your glucose glucose stores have been used up, remember we only have 12 to 24 hours of our glycogen reserves, you're not eating, the only way then to have substantial levels of a hydrophilic circulating fuel is to make these ketone bodies from fat breakdown, and you have months worth of reserves with your adipose stores. Okay. Couple pieces I know I mentioned in class. I just want to make sure to remember them here. They weren't written down here. But talking about certain things that cannot be exported and circulated around the bloodstream. We don't export and circulate ATP. That's something that's used locally within cells. We don't export and circulate acetyl-CoA. So we need to have a mechanism then of circulating sort of equivalents of these. And so ketone bodies are sort of an equivalent way of circulating acetyl-CoA. So beta oxidation is kind of the biochemical name for fatty acid breakdown. So it's a 4-step process occurring in the mitochondria. The flip side is fatty acid synthesis, where you take excess acetyl-CoA. We've talked about this generally, but we haven't really applied a biochemical name to the process. We take and we zip together acetyl-CoAs and we make them into fatty acids. Then those fatty acids are hooked together to make triglycerides. Those are stored in adipose tissue. Highlighting, this process occurs in the cytosol, a really important regulatory mechanism, if you will, for cells to determine what's going on is to differentiate it based on cytosolic versus mitochondrial. And that's highlighted here with what we see with fat metabolism. So a little bit more on ketone bodies. Remember, ketone bodies are basically the zipping together of acetyl-CoA into, in this case, 4-carbon acetoacetate. We talked about ketone bodies earlier in the semester, so just kind of seeing it revisited here. We can export acetoacetate. You cannot export acetyl-CoA. So it's kind of a way of disguising and hiding acetyl-CoA in a way that you can export it. Highlighting, you do not see every cell producing ketone bodies. This is a liver-specific process. And importantly, the liver actually can make ketone bodies, but it doesn't have the mechanisms to break them back down, which is an important safeguard to say that it just doesn't make them and then break them back down again and use themselves. So acetoacetate is the oxidized 4-carbon form. We can see here that beta-hydroxybutyrate is the reduced form. We talked a little bit in class about how to identify and differentiate redox state of molecules. So if we look at this carbon atom, that's the one that's different. When it's carbon as a ketone, so it's doubly bonded to 2 oxygens, you can think about that as having more oxygen versus a carbon here that's just singly bonded to oxygen. So that's an easy way to kind of examine sort of the oxidation state of a fuel metabolite here. An important process here is that when we reduce acetoacetate to beta hydroxybutyrate, we oxidize NADH to NAD+. And then another chemistry that can happen with acetoacetate is we can do decarboxylation chemistry. This is what we call a beta keto acid. Follow me here. This is an acid group, a carboxylic acid group, not at the alpha, but at the beta carbon, we've got a keto group, a ketone. So beta keto acids easily decarboxylate. We'll look more at the mechanism of this next semester, but rapidly or easily decarboxylating generates acetone. So one of the things that can happen is if you have excess levels of these ketone bodies, that can actually be problematic. It can drop your pH. This is what we call ketoacidosis. That can be problematic for diabetic patients. But what happens is your body, in trying to eliminate this excess metabolite, will decarboxylate it and generate acetone. Acetone can be then exhaled, and so that's a way of your body trying to rid itself of something that's accumulating in excess. So we talked about somebody who is maybe in a state of diabetic ketoacidosis, their breath might smell sweet, and that's because they are trying to rid themselves of an excess of these ketone bodies. All right, protein metabolism. Highlighting the main purpose of protein consumption is not for fuel value, but it's actually to generate building blocks that you can reuse. Maybe you're building new proteins, maybe you're converting those amino acids into important amino acid derivatives. We talked about a lot of those in our amino acid and protein learning unit. So when you have, and I mentioned this already, an amino acid, looking at a generic amino acid, right? What differs in every amino acid is what this R group is, ranging from a hydrogen, if it's glycine, to more complex aromatic molecules, right? Or substances there. So when we metabolize amino acids, again, if they're in excess, this is what we'll do. You basically are going to be breaking apart and metabolizing the amine portion here. And we're very just generally saying that goes to the urea cycle. We'll learn details of that next semester. But then you have an alpha keto acid. So this is just basically carbon, hydrogen, oxygen, looks like a carbohydrate derivative. And who you look like kind of feeds into here. And this is intentionally small here because you do not need to know these details, but it's highlighting that different amino acids look like different citric acid cycle intermediates, different branch point metabolites like acetyl-CoA or pyruvate. And it is important to distinguish that whether you are an amino acid that can be made into pyruvate or acetyl-CoA sort of determines your fate. So amino acids that can be made into pyruvate we call glucogenic amino acids. Amino acids that can only be made into acetyl-CoA are called ketogenic, and that's ultimately determining what their fate could be. Remembering that we cannot convert acetyl-CoA back into pyruvate. The only fate for acetyl-CoA derived from those ketogenic amino acids is to convert them into ketone bodies, whereas glucogenic amino acids that can be converted into pyruvate can be siphoned off and used via gluconeogenesis to make glucose. So again, that's what's highlighted right here. So kind of putting this all together with how things like the Atkins diet works, why the ketogenic diet works so well to kind of like eliminate body fat, is if you are not consuming carbohydrates, you still need to maintain 5 millimolar glucose. So if you're not consuming carbohydrates, there's really not a top-down approach. You don't have a lot of glycogen reserves. So what you're going to be doing is using amino acids that are derived either from proteins that you've consumed, or we'll talk about fasting in just a moment. But you basically take those glucogenic amino acids, and once they're made into pyruvate, gluconeogenesis will convert them into glucose. This is a very energetically costly process. So where do you get the energy from it? Well, you get the energy from adipose tissue derived triglycerides. So that's why this whole process works so well. That, and as we'll start touching on in our next learning unit, once you get your hormone levels under control, so learning about endocrinology, eliminating carbohydrates allows you to become more insulin sensitive, and insulin sensitivity is really key to sort of maintaining this fuel partitioning philosophy. We'll get into a lot of that next semester. Just mentioning briefly about fasting, right? If we're considering fasting, that means there's no fuel consumption, no exogenous source of fuels. So you have to maintain 5 millimolar glucose. And so this is really essential for how your body kind of maintains homeostasis, is maintaining that 5 millimolar glucose. So what you need to then do is think about where you're going to get those glucogenic amino acids. And that generally is from the atrophy of selective organs like the liver and the kidney. So if you think about this from an evolutionary standpoint, right? If you haven't been able to find food, not because you're choosing to fast, but because you can't find food, it's important that you not have your muscles atrophy, because you need to still be able to go out and forage and hunt and try to find food. So you atrophy tissues that you can sacrifice a little bit to provide that energy, or those glucogenic amino acids, to provide the glucose that you need for your body to run. All right. Some big picture things here, and then we'll wrap up with the study of pKa. So our fuel equation, fuel plus oxygen gives carbon dioxide, water, and energy. Whether we're talking about burning a fuel or metabolizing a fuel, and we'll get to the sponge analogy in just a second, it's that same basic equation. And these fuels are worth energy because of the electrons which hold the atoms together. So what I want us to think about is the important difference between combustion and metabolism is we are able to harness and save that energy. Okay, so I want you to think about it as an analogy. Let me start over here with combustion. We can squeeze all of the energy, all the electrons out all at once if we burn a fuel, if we combust, for example, a marshmallow here. However, if we imagine fuel as a sponge that contains energy as electrons, and we imagine those electrons being water, metabolism is really squeezing out that water from that sponge, and importantly, collecting that energy to sort of save it. So we can think about the water being electrons, the fuel being our sponge with those electrons, and NADH, again, is going to be something that we can think about, or I should say NAD plus and FAD. Those are going to be things that are going to allow us to save that energy because we're going to reduce those cofactors. Another big picture sort of concept here is if we think about kind of an energy continuum here. We've got fully reduced carbon atoms as methane, fully oxidized carbons as carbon dioxide, and our fuels live in different sort of spaces along this continuum. So fatty acids are more towards the reduced end of the spectrum. So if we think about how much energy can get squeezed out to become carbon dioxide, it's more. That's why they're worth 9 calories per gram. However, if we think about carbohydrates, they're already partially along that oxidation continuum. So that's why they're not worth as much, only 4 calories per gram. All right, just a little bit kind of review here on these electron carriers that I mentioned, right? We've got NAD plus in its oxidized form, its reduced form, NADH. Do want you to remember your B vitamins, so that's vitamin B3 derived, so niacin. This is the redox active portion of this molecule, and what's different with NAD plus versus FAD is it takes on electrons 2 at a time as hydride. So again, 2 electrons are going to come by the addition of hydride. So on the other side of the spectrum is we have a molecule that's responsible for transferring 1 electron at a time. So that's FAD, taking 2 radicals, hydrogen radicals, 1 at a time, that can generate ultimately, when it takes on 2 electrons, FADH2 in its reduced form. So both of these will take a total of 2 electrons, but NADH takes it 2 at a time as hydride, FAD takes it 1 at a time as hydrogen radicals. So again, here's the redox active portion of FADH2. And so this is, again, vitamin B2, riboflavin. You do not need to know structures of these, but I do want you to know the B vitamins that they're derived from and how they transfer electrons. Okay, ATP, another one of our important biological molecules. I want you to remember how much it's worth, that it's our biological money. Right? We generate it by using a proton gradient generated in the electron transport chain that's going to take ADP and combine it with a phosphate to generate ATP. When it happens in that fashion, that is oxidative phosphorylation, using a proton gradient to run an ATP synthesis machine. And again, this requires the coupling of electron transport and oxidative phosphorylation and only occurs in the mitochondria. When we have higher energy compounds, those that are worth more than 30.5 kilojoules per mole that are phosphorylated, they can actually donate their own phosphate to ADP to make ATP. This process is called substrate level phosphorylation. This occurs quickly. It can occur in different areas of the cell, cytosol or mitochondria, but we learned about it generally here with glycolysis. We'll learn next semester, and those of you who are interested in sort of exercise physiology and so forth, anybody who takes creatine supplements is actually a way of creating a hidden reserve of ATP in your body by making a phosphocreatine molecule that can quickly generate ATP by substrate level phosphorylation. So we'll get into that story next semester. A little bit more again on branch point metabolites. Two that I want you to remember right now are pyruvate, that's our 3-carbon branch point metabolite, and then acetyl-CoA is our 2-carbon branch point metabolite. So when we think about these, they're called branch point metabolites because lots of things feed in or out of metabolic pathways at these molecules. That's why we call them branch point metabolites. So acetyl-CoA can be converted into pyruvate by pyruvate dehydrogenase chemistry, reminding ourselves that this is a one-way reaction. Right? So take acetyl-CoA and make it into, or take pyruvate and make it into acetyl-CoA. So this is just kind of a summary slide. We've talked about most of this already. Acetyl-CoA cannot be made into pyruvate. And since fats can be made into acetyl-CoA, but acetyl-CoA cannot be made into pyruvate, and you need pyruvate to make glucose, gluconeogenesis, this means that fats cannot be made into glucose. So kind of a roundabout way to remind us that fats cannot be made into glucose. We've already kind of mentioned this already, but I think this was just a review of this. 3 possible fates for acetyl-CoA. I know this is a homework question that you have, and it depends first on whether you're in a fed state or a need state. And again, if you're in a need state in the liver, we're all considering in the liver here, whether you're helping out locally or globally determines what happens in that need state. So let me just run through and summarize that again. If you are in a need state, your body is in a need state, your liver will say, well, do I need that locally? Am I personally in a need state? If that's the case, I'm going to take acetyl-CoA to the citric acid cycle. I'm going to break it down and take those NADH and FADH2 molecules to the electron transport chain. Oxidative phosphorylation is going to be used to make ATP. However, and simultaneously, this is not an either or, the liver can also help in a global sense. It can take some of that acetyl-CoA that's likely fat-derived, convert it into ketone bodies, and then export it for all other cells to be able to utilize as a global hydrophilic circulating fuel. Flip side, what if you are in a fed state? What if you have excess acetyl-CoA? Remembering that all fuels can ultimately be funneled to acetyl-CoA. Well, then your liver is going to take and reassemble those into fatty acids and triglycerides, export them for storage in your adipose tissue. And as we'll learn a little bit next learning unit and a lot next semester, hormones play a key role in sort of this fuel partitioning pathway as well. All right. Location, right? Real estate agents say location, location, location. Well, biochemists say it too, because where you are really determines the biochemistry that's available for you. So a lot of the chemistry that we're learning about, I should say biochemistry that we're learning about, is ubiquitous, which means it's sort of found throughout living organisms. So whether you are a bacterial prokaryote or an animal or a plant cell, as eukaryotes, a lot of the biochemistry is the same, but sort of the ability to do things might be a little bit different. So when we think about prokaryotes, I want you to again think about it as sort of a giant soup. Everything's kind of all mixed together. Well, we've talked about a lot of processes being differentiated whether you are in the cytosol or the mitochondria. So how do bacteria do that? Well, they don't have those organelles. They don't have mitochondria. So what they do to create sort of a partitioning between a cytosol and a mitochondria is they have an inside and an outside of the cell. So they really use their cell wall to create these partitions and these divisions. And that's different from eukaryotes. So eukaryotes have organelles. Importantly, we've been talking about mitochondria as an important organelle. And differentiating some terminology here, all eukaryotes and having those organelles represent pieces that are within this broth of what we call cytosol. So all the liquid stuff that's inside a cell that's not a specific organelle is called cytosol. And when we mix the broth and the pieces together, that soup is what we call cytoplasm. Okay, so just highlighting a really unique thing here that I think is really cool about plant cells. Animal cells only contain mitochondria, whereas plant cells contain both mitochondria and chloroplasts. And remembering that these are ancient bacteria, or that's the working theory, is to how eukaryotes developed is by engulfing kind of early prokaryotes that had unique sort of abilities. We've got some really fun biochemistry to learn next semester when we study plants. We're not going to spend too much time studying plants, but the fact that they have so many unique organelles in them makes them really unique. All right, we spent a good bit of time on this slide, so I want to make sure to highlight this concept of substrate cycles and flux. So when we think about two substances being interconverted, we can call this a substrate cycle, and what happens in that substrate cycle, what we call the flux or the flow through that pathway in the substrate cycle, is basically the difference between a forward reaction, if we call it converting A to B, and then a reverse reaction converting B back into A. And some big picture things to think about here is if we have a process that is downhill, that gives off energy, that's generally a catabolic process. Okay, and those are going to be happening under need state. If we have a process that is the reverse of that, it's got to require energy. We can't have two processes that both go downhill. These uphill processes requiring energy tend to be our anabolic processes, and they more likely happen in a fed state. Now, that's not always going to be the case. That's not sort of a universal umbrella, but that's generally the case that catabolic processes produce energy and anabolic processes require energy. So if we think sort of generally what happens if we compare these forward and these reverse reactions, if the forward and the reverse reaction rates are equal, we can say that the net flux through this substrate cycle or this pathway would be zero. We're not going in either direction. We're sort of spinning our wheels if the forward and the reverse rates are equal. What more likely happens is one of the pathways is going to be dominant. So for example, if the forward rate is faster than I should say that's going to be this B situation. If the forward rate is faster than the reverse rate, we're going to favor the formation of substance B. And so flux is going to sort of favor that production path of B. Whereas if we're having the reverse rate be faster than the forward rate, in that case, we're going to favor the production of A. So the last thing that we kind of talked about here was just giving you 3 examples. Again, another homework question that I want you to be able to put this in your own words is thinking about, well, how big does this substrate cycle have to be? You know, we can drill down to an enzyme level. So for example, if we consider the enzyme lactate dehydrogenase, we really can consider the interconversion of pyruvate and lactate. So that's a very kind of really in the weeds, if you will, substrate level, substrate cycle, or metabolite level substrate cycle. We can also think about things on a pathway level. So for example, we can think about the interconversion of glucose and pyruvate. Glucose to pyruvate is glycolysis, pyruvate back to glucose is gluconeogenesis. So that's a substrate cycle when we're considering entire metabolic pathways. And then we can even think about an atom cycle, right? And this is really the carbon cycle. And so we can think about glucose going all the way to carbon dioxide and water, and then carbon dioxide and water going back to glucose. And we don't have the ability to operate a substrate cycle in that way because that forward direction is fuel metabolism. The reverse is photosynthesis. And obviously we cannot do photosynthesis. So big picture concepts to think about how we think about interconverting metabolites and how close they are to one another in a biochemical system. So just a fun little picture here to remind us that, again, we've mentioned this a couple of times, we can't go downhill both ways. A neat little MC Escher drawing here that shows water that appears to be flowing downhill both ways. Well, we can't have one of those sort of perpetual motion machines, right? We can't be downhill both ways. And what we tend to see is one pathway is going to be downhill in energy, and the other pathway will utilize different enzymes and will require the input of energy. So that's going to be a good reminder for us for some different processes that we'll study next semester. So the last thing we had before we delved into our pKa story is thinking about the difference between near and far equilibrium reactions in enzymes. Big picture, if something is near equilibrium, that enzyme operates with a delta G close to 0. Far equilibrium operates with a delta G that's large and negative. Okay, so we had two toy analogies to help us think about how this works. The seesaw really can be easily maneuvered by putting weight on one side or the other. And so the idea with a near equilibrium pathway or enzyme is that that enzyme will readily reverse direction depending on whether you're product heavy or reactant heavy. And so that doesn't represent a good control point for flux in a metabolic pathway because you can easily kind of change direction. Okay, so it's easily reversible by changing the concentration of metabolites, so it's not going to be a good control point. Thinking about our water flow analogy, this is not a spigot that you can turn on or off. Okay, you're going to have flow of water through that enzyme depending on which way you are heavy. So not a good way to control flux through that pathway. We are going to be looking for good control points for metabolic pathways by looking for enzymes that are far equilibrium. They are enzymes that have large negative delta G values. How this happens is that enzyme really has a spigot on it. We can turn it on or off. And when you pinch a pathway and you turn off an enzyme, okay, that allows you to build up stuff behind it and get rid of stuff that's downstream of it. And that building up of pressure, if you will, we can think about it like a slingshot, is really what creates this large negative delta G. And that large negative delta G really is because where you want to be, that K, Q versus K analogy when we talked about sort of thermodynamics, Q represents where you're at and K represents where you want to be. How far apart those are from one another sort of determines your thermodynamic driving force, your potential, the magnitude of your delta G. So at the end of the day, the enzymes that we're going to spend time studying are ones that have large negative delta Gs because we can turn those enzymes on and off, and in doing so, we can control flux through pathways. All right, last little piece here is thinking about pKa.